Have you ever experienced a time when you walked in the park and saw three beautiful spiders? Let's call them Spider A, Spider B, and Spider C. I bet the first thing that will come in mind is that you will assume they were the same species, right? Because their coloration and body shapes looked alike. Well, too bad. Unfortunately, they did not belong to the same species. Identifying spider just by looking at the color and body shape is not really an accurate way to tell the species because many spiders share similar appearances across different species. If we look carefully at spider A, it appears to be a different species due to its distinct coloration. However, if we look at spider B and spider C, they might belong to the same species due to their shared coloration. Well, again, they are not. Not unless you are an expert in arachnology. So, how to overcome this problem for non-experts like us? Well, the answer is to look at the spider DNA. Our research group at Spider Bio Unilab, University Alam Campus has engaged in the s5H or Spider 500 project, in which we aim to discover as many as 500 spider species in Perlis within 10 years. Using DNA barcoding technology. But what is DNA barcoding? DNA barcoding is the use of fragments of DNA sequences to identify a specific spider species. This DNA fragment is unique for a given species. Although there are many types of DNA sequences that can be used as a marker, we choose to work with cytochrome oxidase 1 or CO1 DNA. We can think of CO1 DNA as an identity card or IC for spider species. Now, spiders can also have their own kad pengenalan. Awesome, isn't it? So how we do it? We first collect the spiders from the wild, extract the genomic DNA from the legs. Then, two short DNA primers, forward and reverse, are designed and eventually used to amplify the target COI gene from the spider's genomic DNA. Once amplified, they are resolved and visualized by the gel electrophoresis. Next, the COI gene is sequenced by Sanger sequencing and analyzed using bioinformatic tools. Once the identity of the spider is verified in the database using the COI gene sequence, it is converted into a barcode or the identity card before stored in the inventory database. Now that we have assigned a DNA barcode or the IC card to the spider, what else can be done? Just by looking at the barcode does not tell us much anything about the spider species. Thus, what we do is that we convert the DNA barcode or the IC card into a 2D QR code using a specific software, and we eventually customize a so-called hybrid DNA QR code, which is a fusion of the DNA sequence with the typical QR code. Using this hybrid DNA QR code, users can simply scan the code using their smartphone that links to the inventory database to obtain the information of the spider species. So, this is how our innovation works. By integrating the DNA barcoding technology and digital advancements, we bring spider identification process one step closer to accurate and efficient documentation, revolutionizing our understanding of spider biodiversity and its importance. It is hoped that our biodigital product can be used in the development of DNA barcode-based big data database for spiders in the near future.